Thank you so much, Christine. I really appreciate the invitation to speak from the organizers. I'm excited to tell you about the work that my lab has been doing in using optical approaches to probe the function of the cortex. So my lab is really interested in understanding how sensory inputs that are received at the periphery are transformed through cortical circuits into neural signals that can represent concrete features of the external world and therefore be used to support perception and guide behavior. And one of the most famous such representations is of orientation. And this is actually briefly described by Na in her talk earlier, which was originally observed by Nobel laureates Hubel and Wiesel when they were recording from cat visual cortex in the 1950s. And what they found is that when they presented bars of light of a given orientation, that they could drive a neuron very well to cause it to fire all these spikes or action potentials. But if they changed that orientation of that bar even slightly, that they got many fewer spikes or even no spikes at all. And so this was really exciting as being kind of one of the first representations to be seen in the cortex of something, of a really concrete feature in the external world. And since that time, it's become clear that this is a really ubiquitous representation that has been seen in basically every species that has a cortex, ranging from humans all the way most recently to the armadillo. And so not only is this a ubiquitous representation across species, but it's also seems to be broadly represented across brain areas. And so this is true in the monkey, but also in the mouse visual system, which is the species that we use to investigate cortical function because of its experimental and genetic tractability. And so this is a map looking down on the surface of the visual cortex where you see primary visual cortex. And in this case, these authors have investigated or have mapped out the different higher visual areas that receive inputs from the primary visual cortex. So these higher visual areas, these clusters of axons that receive direct input from V1. And so interestingly, despite these each being kind of like their own island of information processing, that recent work has shown that the way that they process orientation seems to be remarkably similar. So this group, Ed Calloway's group, used two-photon imaging to determine the orientation selectivity of neurons across all of these different areas. What they found was that the orientation selectivity was remarkably similar. There were some variants and maybe some areas are slightly more orientation selective, but overall, the take-home message is that all these areas do seem to robustly encode orientation, which to me begs the question, which one of these areas, or if any of them, is really necessary for the perception of orientation. And so this is the question that we're going to address today, in particular to determine whether these four areas, V1, primary visual cortex, lateral medial area, anterolateral area, or posterior medial area, is necessary for the perception of orientation. And we chose these areas because these three higher visual areas receive very strong input from primary visual cortex, and they're well characterized and known to strongly represent these orientation signals. So there's a number of possible outcomes of what we could find. And I think it's important to appreciate that one of those outcomes could be that none of these higher visual areas really are necessary at all for the perception of orientation. We know that orientation is robustly encoded in the primary visual cortex, and that it projects directly to decision-making areas like the colliculus, the striatum, the pons. And so it could be that all this information just shortcuts all of this extra processing and goes right to the output. Alternatively, we might imagine a hierarchical structure where V1 projects to LM, which is kind of a V2-like area, which then sends its information to these decision-making areas. Alternatively, there could be this divergent input both to LM and AL, or you could have a fully distributed network where this information essentially is both present and used across the entire brain. And so in order to get at this question of which of these areas are really important for this computation and this perception, we're going to go through and serially silence each of these areas. And the approach that we're going to use is an optogenetic one. So this is going to take advantage of the tools that Ed Boyden described to us yesterday, channel rhodopsin being a very common one, where these are light-gated ion channels that when activated cause depolarizations in these neurons and can be used to spike very precisely. However, we want to silence cortical areas. We don't want to drive them. And so we're going to take advantage of a sign inversion in the cortex. We're going to express this channelodopsin in the membrane of inhibitory interneurons. And then what we can do is with precision, activate these inhibitory interneurons and thereby silence the local pyramidal cells. And so this can give us both spatial and temporal precision in our ability to silence these cortical areas. So the way that we identify these cortical areas in vivo is by implanting a cranial window, kind of again like Na spoke about earlier, over the cortex, and this can then reveal V1 and these higher visual areas, and we can identify their locations by using intrinsic signaling imaging approaches. And so there are fluorescent proteins that are actually endogenously expressed in the mitochondria of neurons. And these fluorescent proteins actually increase their fluorescence when neurons become more active. And so when we present a visual stimulus, this drives these neurons to fire, and therefore you can see these increases, these brightenings here are the result of the firing of these neurons from these intrinsic signals in the brain. And so then we can use these to target viral injections to express channelodopsin in the cortex, here in this case in LM and in PM, specifically in populations of inhibitory interneurons. In this case, it's parvalbumin positive neurons. And so we can test the efficacy of this approach by using extracellular recordings. So these are probes that have 4 shanks. One shank will stick right in the center of one of these injection sites, and then the other 4 shanks are separated each by 400 microns. And so we can look at increasing distances away from that injection site to see what the influence is in space and in time. So here is some neurons from one of our recordings. These neurons actually were excited by our light, and therefore you can see that they increase their firing rate when the light is on, and therefore these are likely these parvalbumin positive interneurons that are going to be inhibiting the local pyramidal cells. And indeed, the majority of neurons that we recorded from were strongly suppressed by this manipulation, and notably that they were suppressed during the whole duration with which the light was on. And so this gives us kind of a long period during behavior in which we can reliably suppress neurons. To measure the effect across distance, we can then divide this population of neurons according to where it was relative to our injection site. And indeed, we find that neurons that are in the injection site were suppressed to about 60% by about 60%, whereas neurons, as you get further and further away, were much less suppressed by our manipulation. And so that gives us a space constant of about 500 microns, which you can see from this is on the order of the size of one of these higher visual areas. And so we were able to have then the spatial resolution to independently silence these areas. Okay, so now we have the methods by which we can manipulate them. And so now the last thing we need is a behavioral task. And so this is our orientation discrimination task that we've trained our mice to perform. The way this works is they're head fixed, they're facing this LCD screen, they have their paw on a lever, and then a juice tube goes to their mouth. And their job is to push that lever down to initiate a trial, which is going to begin the brief presentation of multiple vertical gratings. And then their job is to detect when a non-vertical grating appears and to release the lever to get their juice reward. So here's a movie of the mouse doing this task. So there he released the lever in response to that target and got his reward. That was a hit. But there, in that case, he released the lever in response to a distractor, which he presumably thought was a target, which we would call a false alarm. So here's just another schematized illustration of this task where the animal pushes the lever down. We have these distractors. He needs to release the lever within a short window following targets. And then on randomly interleaved trials, we can shine the blue light on a particular higher visual area or primary visual cortex in order to determine what the effect is on the animal's behavior. So here is some example data from just one mouse. On the x-axis is the orientation of the target. So when those orientations are large, that means it's a very big change from the distractor. 90 degrees would be as far away as you could get. And when that occurs, the mouse is really good at detecting those targets. His hit rate is close to 100%. And then as those targets become closer to the distractor, his hit rate falls off. And so we can use this to measure the animal's detection threshold. And then notably on these interleaved trials, when we suppress primary visual cortex, the animal does much worse at the task. He's much less likely to detect targets and his threshold is increased. Similarly, we also see that the mouse is less likely to have false alarms, to think that one of these distractors is a target. So that was, this is just example data from a single mouse. Here is data from many mice as we're silencing all of these different, each of these different areas. And indeed what we find is if we suppress V1, LM, or AL, we get a robust increase in these animals' thresholds, suggesting that all three of these areas are really important for the animal to be doing this task. However, suppression of PM doesn't seem to have any effect at all. Similarly, if we look at the false alarm rate, again, we see that suppression of V1, LM, and AL are all reduce the animal's false alarm rate while there's no effect of PM. So one possibility is that this manipulation that we're making really has nothing to do with the animal's ability to perceive the stimulus. It simply is just distracting the animal, causing some general change in arousal. And so in order to determine whether there might be some general effect on the animal's behavior, what we did was move the visual stimulus outside of the receptive field of this population of neurons. And so that means now that the population that we're affecting is no longer the same population that should be encoding the visual stimulus. And indeed, when we do this, we no longer see any effect on the animal's threshold or any effect on the animal's false alarm rate, suggesting that these effects that we're having here on the animal's behavior really seem to be due to the perception of this visual stimulus, and that we're affecting the neurons that are encoding the features that the animal is using for this behavior. So just as a brief summary, what I've shown you is that suppression of primary visual cortex and these two higher visual areas, LM and AL, are all sufficient to impair orientation discrimination. And this is not, I don't think, a trivial result, as I told you that it could be that all of the signals that are present in V1 may have been sufficient for the animal to do this task. And it suggests that the information may be taking some kind of pathway like this, for instance, through the higher visual areas. Interestingly, we didn't find any role in this behavior for this area PM, which was surprising given that there is information about the orientation of the visual stimulus encoded in this area. And so this really made us wonder, well, what is there a different role for PM? And does it have a role in perception? And so in order to test this, we used a different paradigm where we now ask the animal, rather than to detect changes in orientation, we just ask the animal to detect changes in contrast. And we thought maybe this more simple paradigm might be something that would be important for area PM to be involved in. And so now, rather than there being distractors that are presented, the mouse simply is looking at a mean luminance screen and then has to just detect the occurrence of any increase in contrast. And again, we're going to be randomly suppressing the visual cortex on interleaved trials. And so here, again, are some psychometric functions looking at this behavior. So on the x-axis is the contrast of that stimulus. So when that contrast is high, the animal's really good at detecting that stimulus. And as that contrast becomes lower and lower, he's less good at it. And then again, when we suppress primary visual cortex, we see this robust increase in the animal's threshold, suggesting that, at least in this mouse, V1 is necessary for this task. And again, we see some smaller but still significant decrease in the animal's false alarm rate when we probe that. So again, this is one example mouse, and here's our population of animals across the visual areas, and this actually looks remarkably similar to what we saw in the orientation discrimination task, where when we suppress V1, LM, and AL, we see this robust increase in the animal's threshold, but no change in the animal's behavior when we suppress PM. But interestingly, now when we look at changes in false alarm rate, we still see some reduction in the animal's false alarm rate when we suppress V1, but now there's no change in the animal's false alarm rate with LM and AL, and interestingly, an increase in the animal's false alarm rate when we suppress PM. And so again, we wanted to know whether these effects, both on the threshold and on the false alarm rate, were due to perceptual effects or maybe more global changes. And so again, what we did was we moved the visual stimulus outside of the receptive field of these neurons and asked how this affected the threshold and false alarm rate. And so now again, all of these effects on threshold are completely abolished, that these are no longer, that our manipulation no longer affects the animal's ability to detect changes in this contrast. However, While the effects on false alarm rate in V1 were abolished, we still see this trend towards a strong increase in false alarm rate in PM, despite the fact that we've now moved the visual stimulus outside of its receptive fields. And this suggests to us that this increase in false alarm rate might actually have something to do with more broad non-perceptual engagement in the task, potentially something to do with its ability to make the decision. And so in summary, what I've shown is that this lateral network here, V1, LM, and AL, seem like it's really important for perception of both orientation and contrast, and that it modulates the decision-making circuits in this positive way. So when you take it away, you no longer are as good at detecting stimuli. In contrast, it doesn't seem like PM is actually required for any perception of any of these features, despite encoding all of them, which I think is kind of an interesting idea that potentially suggests that there might be kind of some suboptimality to this behavior, and that it's not taking advantage of information that it could be. In addition, we do see this interesting effect on the animal's false alarm rate, which suggests to us that it might have a non-perceptual role in behavior, that maybe, for instance, it might be having some tonic inhibitory effect on the decision-making circuit. And then when we remove PM from the animal's from the behavior, that then we get this disinhibitory effect on the decision-making process. Okay, well, thank you all for your attention, as well as I want to acknowledge Meow Meow Jin, who is a previous graduate student in the lab who did all of these behavioral experiments, and my funding sources as well.